This is the longest lecture video in the series, but it covers a lot of important topics. So let's get started. A factor is an independent variable that the experimenter is controlling. In fact, factor and independent variables are two words for the same thing. It's the thing that the experimenter is testing for. Now, factor level is the values that that factor takes on when it is applied to the experimental units. We also call factor levels treatments. So some examples, going back to this prolac effect on milk production in dairy cows. So prolac would be the factor. And let's say I gave cows either 0, 50, or 100 milliliters supplementation. So those would be my factor levels. I'd have one factor, the prolac, and then three factor levels, 0, 50, and 100. Similarly, the effect of boron fertilizer on canola yield. So boron is my factor, and then the specific rates I'm applying to my plots, 0, 100, 200, 0, 100, 200, and 300, those are my four factor levels. Now, in experimental studies, we have the ability to apply treatments. This is different than in observational studies where we can't apply treatments. We just have to observe, collect data via observation, and then try to make correlations or inferences. We're not applying treatments. Now, the pattern in which we apply treatments to the experimental units is called the experimental design. And this is a very important aspect of the scientific method because this experimental design determines what kinds of statistical tests we can do and the statistical power of our treatment comparisons. Regardless of the experimental design that we choose to go with, we're always going to be randomly assigning our factor levels or treatments across the experimental units. At the start of the experiment, each experimental unit has an equal chance of receiving every single factor level, in other words. Now, why is this so important? Remember what I said earlier in a previous lecture, that what randomization lets us do is account for unknown underlying sources of variation or error that can confound our experiment. A classic example is if we're doing an agronomy experiment in a field, by randomly assigning treatments, we can minimize the effect of changes in soil texture or soil fertility, for example, that some parts of the field are just higher yielding or lower yielding than other parts. Now, when we randomize, the key here, now almost all of the statistical tests that we're going to use in this course, and really in most of the statistical tests we do as scientists, they assume that the error, the random underlying error, is the same across experimental units. And if we randomize, that is almost always true, that each experimental unit has the same amount of error or unknown underlying variation than any other experimental unit in the study. That's true if we randomize. It is not true if we do not randomize. If we decide, at the north part of the field, I'm going to put all my low-end rates. At the south part of the field, I'm going to put all my high-end rates, or whatever. Okay, we need to randomize for us to be able to meet the assumptions of many of the statistical tests that we're going to be using. When choosing an experimental design, we have to balance different considerations. Practical ones like the cost, time, and effort it would take to carry out that experimental design. We also want designs that are going to minimize confounding or unaccounted for variation. And especially when we have factorial experiments, I'm going to explain what that is later, but when you have factorial experiments, the statistical power of our comparisons and the types of statistical tests we can do is going to be directly related to our experimental design. So these are all considerations we need to think about. The completely randomized design is the simplest experimental design in the book, and it's a very good one. Basically, for all the experimental units in the study, treatments are just randomly assigned. It's very simple, very straightforward. The benefit is that we have very high degrees of freedom, and that's good because, again, higher degrees of freedom means more confidence in our results. A drawback is that we can't block for known sources of variation. So here's some example of completely randomized designs. Let's say we want to look at the effect of 4 different diets, feed types, and their effect on weight gain in pigs. So let's say we have 16 pigs. That's how many pigs we have. We have 16 experimental units. We have 4 treatments, or I would say factor levels, but we have one factor, which is the effective diet. And we also have 4 replicates. So, 16 experimental units divided into 4 treatments, 4 replicates of each treatment. Now let's look at the degrees of freedom and how we partition the degrees of freedom. So df is equal to degrees of freedom, is shorthand. And when we look at the total degrees of freedom, it's our total sample size, or experimental unit number minus 1. So total degrees of freedom is 15. And we partition these degrees of freedom between our treatments and our error sources of variation. So for degrees of freedom of treatments, we have 3. And because we have 4 treatments, and so treatment degrees of freedom is the number of treatments minus 1. And then we have our error variation, which is just our total degrees of freedom minus the number of degrees of freedom of our treatments. So 15 minus 3 equal to 12. Notice here that in the completely randomized block design, we're not accounting for factors that might influence weight gain. Let's say, we have 16 pigs, but 4 of them are in the north side of the barn, and in the north side of the barn you gain weight more slowly than the south side because you're closer or further away from the feed trough or whatever. We're not blocking for potentially confounding variables. Here's a field example of a completely randomized design. This is a photo of the Winchester Agricultural Research Station in Eastern Ontario, and you can see I have 12 plots. I've just made this up a little bit. I have 12 plots, and I have 3 different rates of fertilizer I'm applying. So I have 1 factor, which is fertilizer. I have 3 factor levels, or 3 treatments, and I have 12 plots. So I basically have 4 replicates because I have 12 plots, 3 treatments. So each treatment is applied to 4 different experimental units. And in a completely randomized design, this is what it would look like if I just completely randomly assigned my treatments to my experimental units. Now just notice, unfortunately, what happened in this completely randomized design, that my treatment number 3, 100 pounds per acre, was all in the, let's call this the south end of the field, whereas almost all of my treatment 1s and 2s were in the north end of the field. Now, this just happened randomly, but this would be very common in a CRD, or a completely randomized design, that your treatments clump up. Now, if there's variation across this field, say the north side is more fertile or less fertile than the south side, then I'm going to have lots of error, and I won't be able to have very precise measurements or very precise treatment effects. And so in this case, blocking is going to be very advantageous. Looking at the degrees of freedom across my sources of variation, I have 12 plots, or experimental units, 3 treatments, and 4 replicates. So my total degrees of freedom in the experiment is equal to 12, or experimental units minus 1. My total treatment source, degrees of freedom for my treatment source of variation is 2, because I have 3 treatments, and so 3 minus 1. And then my error, the degrees of freedom for my error term, is 9, which is just 11 minus 2. A randomized complete block design is used when you're certain there will be variability that you can block against. Now, the complete in the randomized complete block design title means that all the treatments are found within a single block. Now, you can have incomplete block designs where, let's say you have 10 treatments, but some blocks might only have 4 treatments. So we're not going to worry about incomplete block designs. There's reasons why some people use them. But RCBDs are very common. Now, the benefits of RCBDs are that you can better account for unknown variability via blocking. You can also deal with missing plots more easily. I won't get into the technical details here, but you can replace missing values if you wanted to much more easily in an RCBD. Now, a drawback is that you're going to be using up degrees of freedom with blocking. And so the confidence we can have in some of your other sources of variation, or the confidence we have in the estimates of those sources of variation, is going to be a bit lower. Let's revisit the experiment looking at the effect of different feed types or diets on weight gain in pigs. In this situation, we're going to be blocking by location in the barn because their location may affect their eating habits. So we have, again, still 16 pigs. We still have 4 treatments, which is the different diets or feed types. And now we have 4 complete blocks, which we can also call replicates. And in each replicate, or in each block, north, south, east, and west, we're going to have 4 pigs, and each pig is going to have one of those 4 treatments applied. So looking at our degrees of freedom partitioning across our sources of variation, our total degrees of freedom here is equal to 15. It's basically the number of treatments times the number of blocks minus 1, which is the same as saying the number of experimental units minus 1, which is 15. Our treatment and block degrees of freedom are both equal to 3. It's just the number of treatments or blocks minus 1. And then the error term is going to be multiplying the treatment and the block degrees of freedom together. And you sum all those up, treatment, block, and error degrees of freedom, and it will equal total degrees of freedom. Revisiting our field experiment, this is now a randomized complete block design with 4 blocks or replicates. And you can see each of the 4 replicates separated by those red horizontal lines. Notice that in each of those between each of those red horizontal lines in each block, I have treatments 1, 2, and 3. I don't have them clumped up in the north or the south anymore, but they're much more evenly spread out across this field. And hence, I'm much better able to block against any random variation that could be running from north to south. Looking at our sources of variation and partitioning degrees of freedom, you can see that our total degrees of freedom hasn't changed. Our treatment degrees of freedom hasn't changed. We now have a blocking degrees of freedom, which is equal to 3, or the number of blocks minus 1. And now you can see our error degrees of freedom is equal to 6, which is less than it used to be in the completely randomized block design. So this can reduce the confidence we have in our estimate of the error variance. But if we block correctly, we're overall going to increase the statistical power of our experiment. Sometimes your block effect will be significant, meaning that the block itself had a significant effect on your dependent variable. For example, maybe pigs in the north side of the barn have significantly higher weight gain than pigs in the south side, east side, or west side of the barn, or that plots in the northernmost block have significant higher yields than plots in the southernmost block. And so your block is going to have a significant effect. Now that's good. It means that you're accounting for this variation and partitioning it to your block effect as opposed to partitioning it to your error and therefore probably washing out and having no significant mean effect. So it's a good thing that you have a significant block effect. But if block effects are very, very large and significant, then you often need to go into your statistical model and modify the error structure. So you have to normally change to a heterogeneous error structure. Anyway, you don't need to know the details of that at this point in your career, but a point to take away right now is that if you expect very large block effects at the start, it's better to exclude the area in the first place. So, for example, if I knew that pigs gained way more weight in the north side of the barn, I would just not have my pigs in that area. I would exclude that area. Or if I knew that yields were much higher in the north side of the field, for example, that area of the field had lots of manure applied in its, decades earlier, I would not block against that. I would just exclude the whole north side of the field and just focus all my plots, put them all in the south side. So better to exclude areas if you expect large block effects. Split plot designs are an option for factorial experiments. Factorial basically just means that we have an experiment where we have 2 or more factors that we're investigating at the same time. Now, if one of those factors is really impractical to randomize at the scale of the experimental unit, then we can apply one variable, that hard to randomize factor, as the main plot factor, and randomize that main plot factor within each replication. And then within each main plot, we randomize the second split plot factor within that main plot factor. So here's an example, a classic example. Let me move forward here. A classic example would be a classic example of a split plot field experiment would be anything involving a tillage factor and different tillage treatments, because they're very hard to implement at the level of the experimental unit or small plot. By the time you have the tractor at the right speed and the tillage unit engaged and working the soil properly, you're already 3/4 of the way down your plot. So you're only actually tilling like 1/4 of your plot. So it's very hard to do small plot tillage research. So this is a prime candidate for split plot experiment. So here we have a tillage by nitrogen rate experiment. This is just say one replicate that you're seeing here, the field in front of you. We have 3 tillage treatments or 3 tillage factor levels, whatever those are. But you see we have the 3 strips. We can say that all 3 of those strips, the same type of tillage was applied. And then across each strip, we're going to randomly assign to our plots our nitrogen rate treatments. So again, we have 12 plots, but we have, you see 3 tillage strips, and within each tillage strip, we have one of 4 nitrogen rates applied. So the tillage is the main plot, the nitrogen rate is the split plot factor. Split plot designs are very advantageous because they make certain experiments much more practical, especially when some factors are very hard to apply to the experimental units. A disadvantage is that you lose degrees of freedom when you're comparing your main plot factor, but you gain degrees of freedom for comparing your split plot factor. So there's a bit of a trade-off here. Now, what does this mean? Going back to that example, the tillage by nitrogen rate trial, I'm going to have more statistical power comparing or looking for significant differences between my nitrogen rate treatments compared to significant differences between my tillage treatments. I'll have less statistical power looking at differences across my tillage. Here's another real-world example of a split plot experiment. So this was a planting date by soybean variety trial that was done. What we can see here is that I have 4 replicates, and those replicates are, between each of those red lines here. So I have 4 replicates in total. And in each replicate, I have a main plot, which is the planting date, because you can see I planted in either April or May. And within each planting date, I planted 5 soybean varieties. So I have one of 5 soybean varieties in every planting date, 2 planting dates in every replicate, 4 replicates, so 40 experimental units in total. And by the way, you see those little holes in each plot. Those are biomass sampling. So the graduate student, Seth Ritzma, took biomass samples in each of those plots. So looking at the partitioning of the degrees of freedom, the total degrees of freedom is 39 because we have 40 experimental units minus 1. We have 4 blocks, so B minus 1. We have 3 degrees of freedom. We have 2 planting dates, so our degrees of freedom for planting date is 1. We have a block by planting date interaction, so we have to partition degrees of freedom to that interaction, and that is 3 degrees of freedom. We have 5 varieties, so our degrees of freedom for variety factor is 4. We have a planting date by variety interaction. I'm going to talk about that in a minute. That's also 4 degrees of freedom. And then the remainder are error degrees of freedom, 24. So that's the partitioning for a split plot experiment. We can generalize the split plot experiment to more than just 2 factors. We can generalize it to 3 factors. That would be the split plot experiment. We can generalize it to 4 factors, the split plot experiment, and so on. So again, I want to give you an example of a real agronomy experiment, something that a more professional scientist would do in the field, because we're paid to do complicated experiments mostly, not normally simple experiments. All right, so this is a split plot experiment that's going on at 5 locations, but it's the same experimental design at each location, at each research station or farm field. We have 3 different nitrogen rates, and that's the main plot factor. And we have 3 factor levels because we have 3 different nitrogen rates. For each nitrogen rate, we have 6 different ways of applying that nitrogen. We call it nitrogen application method. So we have 6 factor levels for the nitrogen application method. And the split plot, the third factor we're investigating is corn variety. And that there's 2 factor levels because we have 2 different corn varieties. And finally, we have 4 replicates. And when you do the math, that's 144 plots at each location. Let me show you how we lay this out. Let me show you how this split plot experiment is laid out. This is a drone image of the experiment taken by Professor John Sulik with his drone. And the experiment I'm describing, the 144 plots, is in the main experiment. So I've divided the field into 4, representing the 4 replicates, because we have 4 replicates in this study. And let's just look at the replicate on the top left. Here you can see each of the 3 main plot factors. So the same nitrogen rate was applied essentially east to west across your screen, 3 different nitrogen rates. So we have our main plots, our 3 main plots in each replicate. Now I have my split plot in each replicate, and you can see here, this is actually 2 experimental units in each main plot. So if you look here, this is the nitrogen application method that was applied. And then what we have here, this yellow line that you see is the split plot, which is the two different varieties within each application, nitrogen application method. And so if you look, if you look at the replicate, there are 36 experimental units or 36 plots. They're equally divided, but between the three main plots. They're equally divided between the six split plots, and they're equally divided furthermore into the two split plot treatments, which is the two corn varieties. I earlier mentioned factorial experiments. Let me give some more details here. A factorial experiment is whenever you have two or more factors in a single experiment, and each with two or more levels. So to give you a handle on the lingo here, we can talk about a 2 by 3 factorial, which means you have two factors. The first factor has 2 factor levels, and the second factor has 3 factor levels. Like if you're baking cookies and you want to look at the effect of 2 different baking temperatures and 3 different baking times. Now, when you have a 2 by 3 factorial, it means that you're testing all 3 bake times at both temperatures. In other words, you're going to have 6 treatments at a minimum, and that's just with one replicate. So 6 treatments for each replicate. A 2 by 3 by 2 factorial, for example, if you have 3 factors, one with 2 levels, one with 3 levels, another with 2 levels, that means you're going to have 12 treatments, or you need a minimum of 12 experimental units for one replicate. So factorials get really big really fast, okay? Now, regardless of what type of experiment the factorial is, you can apply different experimental designs to it. So factorial experiments can be CRDs, RCBDs, split plot, and there's many others. I didn't go over all the possible experimental designs here. But what factorial experiments do is they introduce interactions. When we have interactions, we refer to main effects and then interaction effects. So main effect refers to the effect of a single factor on the dependent variable averaged over all other factors. So let's say we're doing a nitrogen fertilizer experiment by corn variety. So we have different rates of fertilizer that we're applying to different varieties of corn. And because this is a factorial, all corn varieties are getting all the different rates of nitrogen fertilizer. Okay, so we can say as a main effect, the effect of nitrogen fertilizer increased corn yields by 50 bushels per acre on average. So that is a main effect of nitrogen fertilizer on corn yield, because we're averaging over all the other factor levels, all the other corn varieties. But what if one factor depends on the other? So let's say if we looked at interactions, we could say nitrogen fertilizer increased yield by 150 bushels for the corn variety Triumph and decreased yield for the corn variety Blaze by 50 bushels per acre. So now we have an interaction because the main effect of nitrogen fertilizer, the effect of nitrogen fertilizer on our dependent variable, which is corn yield here, is going to depend on the other factor, which is corn variety. And if we just present the main effect, that nitrogen fertilizer increased yields by an average of 50 bushels per acre, we're actually going to be completely misleading our readers because it actually increased yields by a lot in one variety and decreased yields in the other variety. So when we're presenting our results and talking about significant effects, we always distinguish between significant main effects, so the effect of the factors we are imposing, our independent variables, and the significance of interactions between our factors or between main effects. And both can be statistically significant or non-significant. There are 2 kinds of interactions. The first would be a crossover interaction, what we also call interactions of directions. And this is when a treatment has a positive effect in the presence of one level of the other factor, but a negative effect in the presence of another level of the other factor. And so the graph below, or what you're seeing here, is an example. So in this example, we have the rate of herbicide application on the x-axis and yield of soybeans on the y-axis, and we have 2 soybean varieties. And we can see that on average, or not on average, but the effect of herbicide rate on our variable of interest, which is yield, depends on the variety. Not only does it depend on the variety, but we actually have a crossover interaction that it positively affects yield with variety 1, but negatively affects yield with variety 2. And it's these kinds of interactions that are often significant when we do analyses of variance, And it's these ones we really have to worry about. Because if we just present the main effect of rate of herbicide, ignoring the variety effect, ignoring the interaction, then we're going to mislead our readers. The second kind of interaction are interactions of magnitude, and they're not as serious as crossovers. Basically, what this does is the main factor will always have a significant positive or negative effect on the dependent variable, the variable of interest. So here, this would be soybean yield. But the magnitude of the effect, how strongly positive or how strongly negative that factor influences, is going to depend on the level of the other factor as well. So here, in this example, we have the rate of herbicide application as well as soybean yield. But in this case, the rate of herbicide application increases yield for both the varieties. It just increases it a lot more in variety A than variety B. But we would not be misleading readers if we said, herbicide applications increases yield, because it does it for both varieties. Rules for dealing with interactions when we have factorial experiments. Typically, you are going to first test the significance of the interactions before your main effects. So in the previous example, I would have first looked for significant interactions between variety and herbicide rate. Now, if they're not significant, that's fine. You can proceed as normal to just looking at the significance of the main effects. If they are significant, you have to determine if the interaction is one of magnitude or one of crossover or i.e. direction. So if it's just an interaction of magnitude, normally what I do is, I would state that in the written part of my report or my peer-reviewed manuscript that, there was an interaction, but it was just of direction. And in my table of the data I would just present the main effects. It's different if there's a crossover interaction or interaction of direction. You can't just present the main effects because it can be misleading, as we already talked about. So you would have to present, the interaction of both factors. This is an example of how to present data when you have a significant crossover interaction. So on the left would be assuming no significant crossover interaction in a nitrogen rate by corn variety trial. I'm just presenting the averages of the nitrogen rate. On the right, you see there is a crossover effect. And so I'm presenting the means separately for all levels of the corn variety factor, variety A and variety B, because in variety A, nitrogen rate is increasing yield. And in variety B, as nitrogen rate goes up, yield is going down in variety B. In the experimental approaches that we're going to learn about, there's always at least two treatments, and one of those treatments is classified as the control. Now, the purpose of the control is to provide a direct contrast to the treatment of interest. There's different kinds of controls possible. Sometimes you need multiple controls, but negative controls is the most common. It represents the case of no intervention or the status quo or no effect, and it's usually related to the null hypothesis in a really obvious way. So, for example, in the last experiment example, the nitrogen rate by corn variety trial, my negative control was 0 kilograms of nitrogen unfertilized, because I was looking at the effect of nitrogen fertilizer. So I wanted a negative control that would be no nitrogen fertilizer. Now, we can think of other examples. Let's say we wanted to study the effect of different diets or dietary change in the weight gain of broiler chickens that are grown or raised for meat. Well, the negative control would just be that the typical standard diet without modification. And because our null hypothesis is that this dietary change, whatever we're modifying, is going to have no effect on weight gain. So to actually make that comparison, we need the typical standard diet. The correct negative control is not always obvious. Just for example, consider a clinical trial to test the effect of a new medicine on something like blood pressure. Now, we would think that the correct negative control is just to have no intervention, right? Just not give them a pill. In the control group, a pill. Don't give them anything. But of course, that's not what is done, because it doesn't control for other aspects of the experimental system that we need to consider. For example, The placebo effect. Normally, we would give people in the control group a sugar pill or a pill that you can't really visually or taste-wise distinguish from the actual pill, and that's going to help us account for any effect that might arise simply from the patient believing they are receiving a treatment. Because sometimes people will feel better when they think they're actually being treated, even though the treatment itself isn't doing anything. It's a psychological effect. So to account from that, the correct control isn't to have no pill at all. It's to give them a sugar pill or placebo. Now, another thing we have to consider here is that if the people taking blood pressure know who is and who isn't getting the real blood pressure medicine, their measurements might be biased if they're, say, taking blood pressure in the way you see here. And so another way to have a negative control would be to double blind the study, which means that the people recording blood pressure don't know if the person, in front of them that they're measuring blood pressure on has gotten the actual medicine or the placebo. We also already looked at the real-life example of an experiment to test for the effects of foliar application of the boron micronutrient to spring canola. In this case, the boron was applied with an adjuvant. So we're testing the combined effects of boron per se, as well as the adjuvant. However, if we want a true negative control, we have to apply boron alone without the adjuvant. And we'd probably also need another negative control, which is just the adjuvant alone without the boron. A positive control is a treatment that is included to test some aspect of the internal validity of the experiment. Basically, it's there to demonstrate that the experiment is in fact capable of revealing treatment effects when they exist. It's not necessarily very interesting in and of itself. Basically, when we have a positive control, we have an expectation, a very clear expectation of what that control is going to do to our dependent variable, our variable of interest. And so if it does what we expect it to do, we can show that data to other people and say, look, the positive control behaved as expected. We can have confidence in the internal validity. Whereas if the positive control does not behave as expected, then we know there's something wrong with the experiment. So let me just give you an example of how positive controls can work. Let's say I had a bunch of dairy manure, and I wanted to see what effect the dairy manure had on phosphorus uptake in corn crop. So let's say I applied different rates of manure to different plots of corn, and I measured phosphorus uptake. I would naturally have a negative control, which would be no manure applied. But I would probably also want a positive control. So think about it. It's possible that the field I'm experimenting on that has these rates of manure is just super saturated with phosphorus already. There's just tons of phosphorus in the soil already. So adding more phosphorus in the form of manure is not going to have any significant effect. And that just has to do with where that experiment was located. Just this field happened to be very high naturally in phosphorus. So how do I measure that? How do I see if this is the case? Well, I would have a positive control where I would actually directly add a lot of phosphorus fertilizer to a couple of plots, and I would measure yield in those plots. And so what I could do is I could compare yield in the unfertilized, no manure, no fertilizer, negative control plots, and I could compare yield in the positive control plots that did not get manure but got a lot of phosphorus fertilizer, inorganic fertilizer. And if there was a positive yield response, there was a difference between negative and positive control yields, I could say, aha, look, this is a field that is responsive to phosphorus. And so I can test the effect of this dairy manure on phosphorus uptake in corn because this is a phosphorus responsive site.